Good morning everyone. Today we discuss factors affecting Hardy-Weinberg equilibrium, a brief account of evolution and evolution of human beings. 5 factors affect the Hardy-Weinberg equilibrium. They are gene flow, genetic recombinations, genetic drift, natural selection, and mutations. These 5 factors affect the Hardy-Weinberg equilibrium. Means if they are operating, change occur in the allelic frequency, change occur in the genotypic frequency. So generally, according to Hardy-Weinberg equilibrium, if the population follows certain rules, no change in the allelic frequency, no change in the genotypic frequency. So these 5 factors bring changes in the allelic frequency and genotypic frequency. If any change occur in the allelic frequency and genotypic frequency, so that population evolved into a new species. So first one is gene flow. Gene flow, also known as gene migration. Gene flow, also known as gene migration. In one population, 2 alleles are present, capital A, small b. In another population, capital T alleles are present. From one population, individuals contain small b migrated to another population. Later, the 2 populations are geographically isolated. So the population donate alleles known as a donor population. The population receive alleles known as recipient population. So allelic frequency and genotypic frequency changes in both donor population and recipient population. Before the donor population transfer the alleles, the frequency of capital A and small b is equal to 1. Before the recipient population receive the allele from donor, the frequency of capital T is 1. Or in simple, you may consider here also capital A, small a. So what is gene flow or gene migration? From one population, individual contain some alleles migrated. After migration, the 2 populations are separated. The population donate alleles known as donor population. The population receive alleles known as recipient population. Before migration, in donor population, one character represented by 2 alleles, capital A, small a. So the frequency of capital A and small a equal to 1. Before receiving the alleles, the frequency of capital A in recent population is 1. So after migration, the frequency of capital A increases. Capital A become 1 means frequency of capital A allele increases. The frequency of small a become 0. In the same manner, in recipient population, before it receives the alleles, the frequency of capital A is 1. After receiving, the frequency of capital A and small a become 1. Change occur in the allelic frequency. Similarly, change occur in the genotypic frequency also. Before it transfer, before the migration, two genotypes present in the donor population. Before the recipient population receive the alleles, it contain only one genotype. After receiving, two genotypes. So, allelic frequency and genotypic frequency changes in both donor population and recipient population. So, due to change in the allelic frequency and genotypic frequency, change occur in the character. So, in course of time, new species evolved from both donor population, recipient population. Once again, gene flow or gene migration. Flow of alleles. So, allele is the alternative form for gene. Flow of alleles known as gene flow or gene migration. After migration of individual contain one type of allele, the two populations are geographically separated. The population donate alleles, donor population. The population receive alleles, recipient population. So, in donor population and recipient population, allelic frequency and genotypic frequency changes. Before the donor population donate alleles, frequency of capital A, small a is equal to 1. After migration, the frequency of capital A is 1, the frequency of small a is 0. Similarly, before the recipient population receive the alleles, the frequency of capital A is 1. Now the frequency after migration, the frequency of capital A plus small a is equal to 1. Change occur in the allelic frequency. Similarly, change occur in the genotypic frequency. Before migration, in recipient population, only one genotype. After migration, two genotypes. In this manner, due to gene flow, change occur in the allelic frequency and genotypic frequency. As a result, new character appear. Next one is gene recombinations. Gene recombination. Recombination means new combination. For example, round and yellow pea plant crossed with wrinkled and green. In F1 generation, you already know round and yellow. In F2 generation, round yellow, round green, wrinkled yellow, wrinkled green, obtained in 9:3:3:1 ratio. Parent combination round yellow, wrinkled green. In F2 generation, round yellow and wrinkled green 9:1. The remaining two are recombination round green, wrinkled yellow. What happened? Change occur in the genotypic frequency. Previously only two genotypes, round yellow, wrinkled green. Now four types of genotypes. Frequencies of alleles also changes. So gene recombinations also bring changes in the, due to recombination, allelic frequency and genotypic frequency changes. As a result, a new species evolved. Genetic recombination. So you already know dihybrid cross example. Round yellow pea plant crossed with green, wrinkled green. In F2 generation, we obtained round yellow, round green, wrinkled yellow, wrinkled green in 9:3:3:1. So round green and wrinkled yellow are the genetic recombination, new combinations. So in parent generation, only two genotypes. In F2 generation, four genotype, 4 phenotypes and 16 genotypes. Change occur in the phenotype, genotype. Change occur in the allelic frequency also. So, due to gene recombination also, change occur in the allelic frequency, genotypic frequency. As a result, change occur in the character. In course of time, new species evolved. Genetic drift. Genetic drift. Variation occur in a small population by chance. Variation occur in a small population by chance. By chance, not by selection, is known as genetic drift. Variation occur in a small population by chance, not by natural selection, is known as genetic drift. Before we discuss, I explain genetic drift with the help of example. You already know Piston Betullaria, scientific name of peppered moth. It occurs in two forms, white color and black color. White color and black color. Before industrialization, white color moths were more in number. After industrialization, black color moths are more in number. Why white color moths were more before industrialization? You already know the bark is white in color. The bark is white in color. The white color moths, bark is white in color. Due to white color background, the birds identify the black color moths before industrialization. So before industrialization, the birds eat away most of the black color moths. That's why before industrialization, white color moths were more. Means before industrialization, nature select the white color moths. After industrialization, the bark become black in color. The birds eat away most of the white color moths. Means after industrialization, nature select the black color moths. So what is the definition? Variation occur in a small population by chance, not by selection, is known as genetic drift. In some industrial areas, today, white color moths are more in number. More in number. In some industrial areas, white color moths are more in number. Why white color moths are more in number? So, in some industrial areas, white color moths are more means against the natural selection. That is genetic drift. Variation occur in a small population by chance, not by selection. In some industrial areas, the white color moths may think our population decreases. Instead, they present on the bark. They may hide under the leaves. So now the birds eat away black with some more efficient, they identify the black color mass and eat away black color mass only. Due to this reason, in some industrial areas, white color mass may be more in number. Or in some industrial areas, eggs of black color mass were destroyed. Due to this reason also, white color mass are more in number. In some industrial areas, white color mass are more in number is an example for genetic drift. So, variation occur in a small population by chance, not by selection, is known as genetic drift. Example for genetic drift, founder effect and bottleneck effect. Founder effect and bottleneck effect. Founder effect. A group of organisms belong to a particular species happen to live in a particular habitat. Due to variation, some organisms fail to stay in the same habitat. They migrated, set up a colony. Later they are geographically isolated. The drifted population means moving population, migrated. The drifted population known as founders. From the founders, a new population evolved. New population evolved. The new population resemble like founders, not to the original population. Or we discuss hypothetical example. A group of 20 rabbits. 16 rabbits contain normal limb and 4 rabbits contain long limbs. Long limbs. Due to presence of long limb, they may feel superior. So that's why they are unable to stay in the same habitat with the same members. So they migrated, means they drifted, move away. Set up a colony. Now they are known as founders. Now the founders are geographically separated from the original population. From the founders, a new population evolved. The new population resemble like founders, not to the original population. In original population, 16 rabbits contain normal limb, 4 rabbits contain long limb. So due to presence of long limb, they may feel superior. They migrated, set up a colony. So all founders contain long limb. From the founders, a new population evolved. All rabbits contain long limbs only. The new population differ from the original population. New population all contain long limbs. Original population contain normal limb. So it is a hypothetical example. So what is founder effect? A group of organisms belong to the same species happen to live in a particular area. Due to genetic variation, some individual failed to stay in the same habitat, so they drifted away, means move away. They are known as founders. From the founders, a new population is evolved. The new population resemble like founders, not to the original population, known as founders effect. Next one is bottleneck effect. Bottleneck means natural calamity. Bottleneck means natural calamity. First we discuss the example. Homozygous Amish population. Amish. So one type of sheep, Amish. heterozygous Amish population. Polydactyla Amish population. Amish, name of the sheep species. It occurs in 3 genotypes: homozygous Amish, heterozygous, polydactyla Amish population. Polydactyla, actually more than 5 toes, here 6 toes, also known as polydactyla condition. Homozygous and heterozygous Amish population easily adapted. They have several favorable variations to live in that habitat. Polydactyl Amish population is struggling, extreme phenotype. So at the time of natural calamity, so earthquake or volcanic eruption or tsunami, at the time of natural calamities in Lancaster in USA, in Lancaster town in USA, homozygous Amish and heterozygous Amish population perished. Only survival are polydactyla Amish. So lucky survivors are polydactyla Amish population. Lucky survivors are polydactyla Amish population. From the lucky survivals, a new population evolved. The new population resemble like lucky survivors, not to the original population. So in Lancaster till today, the Amish population represented by only polydactyla Amish, but in other towns and other cities, in Amish 3 genotypes are present. So in this manner, the Amish population in Lancaster differ from other cities and towns. So that is only one genotype. Instead 3, only one genotype. Change occur in the allelic frequency, change occur in the genotypic frequency. Here also in founder effects, the new population contain one phenotype, a particular type of phenotype and genotype. But in original population, 2 phenotypes, 2 or 3 genotypes. So change occur in the allelic frequency and genotypic frequency. So genetic drift, variation occur in a small population by chance, not by selection, is known as genetic drift. Examples: founder effect and bottleneck effect. Founder effect, a small genetically varied group drifted from the original population, move away from the original population. Now the drifted population geographically separated from the original population. The drifted population known as the founders. From the founders, a new population evolved. The new population resemble like founders, not to the original population. Next one, bottleneck effect. So for example is all red Indians. All red Indians have O positive blood group. Red Indians, native of USA. So actually in human beings, 8 types of blood groups: O positive, O negative, A positive, A negative, B positive, B negative, AB positive, AB negative. So in the original population, 8 blood groups, but red Indians contain homozygous O positive. So they are unable to remain with other human beings in that habitat. They migrated to forest in USA. So they are the founders. All red Indians contain O positive. So the members of red Indians opt their partner from red Indian tribe only. That's why till today red Indian contain only one blood group, that is O positive blood group. In this manner, red Indians are genotypically, phenotypically differ from human beings. So, human beings of USA. Example for founder effect. Similarly, bottleneck effect. Bottleneck means natural calamity. At the time of natural calamity, several animals perish. The lucky survivors are genetically differ from the original population. Why in original population, in this example, 3 genotypes are present, but in lucky survivors only 1 genotype. From the lucky survivors, a new population evolved. The new population resemble like lucky survivors, not to the original population. So, variation occur in a small population by chance, not by selection. Nature never select the polydactyla Amish population, but here by chance, lucky survivors are polydactyla Amish population. So, founder effect and bottleneck effect, example for genetic drift. Variation occur in a small population by chance, not by selection. Now we discuss natural selection. Natural selection is of 3 types: stabilizing selection, directional selection, and disruptive selection. Stabilizing selection. Stabilizing selection. Factors affect Hardy-Weinberg equilibrium. So next one is natural selection. Natural selection never create change in the allelic frequency and genotypic frequency, but nature select the allele and genotypic frequency which is favorable to the nature. So the organisms, the allele or genotype selected by nature known as well-fit. Such genotypes easily overcome the struggle for existence. They reproduce and produce more number of offsprings. So natural selection never bring change in, that means natural selection never create new alleles or new genotypes, but the nature select the allele and genotype favorable to the nature. In this manner, natural selection help in the evolution of new species. So natural selection is of three types: stabilizing selection, directional selection, and disruptive selection. Stabilizing selection. Stabilizing selection is operating in stable environmental condition. Stabilizing selection is operating in stable environmental condition. No change in the environment. At the time, stabilizing selection select the average phenotypes, eliminate the extreme phenotypes. Extreme phenotypes means they are struggling for their survival in that habitat. So existing environment is homogeneous, suitable to the average phenotype, and extreme phenotypes are struggling. So stabilizing selection select the average phenotype, eliminate the extreme phenotype from both the ends. As a result, peak become narrow and higher. Means finally in that population only average phenotype is present. Eliminate the extreme phenotypes. Example, in England, birth weights of human babies were estimated in large scales. In most of the human babies born with 8 pounds, so that is the average phenotype. So nature select the human babies born with 8 pounds. Human babies born with more than 8 pounds is one extreme phenotype. Human babies born with less than 8 pounds is another extreme phenotype. So stabilizing selection eliminate. So mortality rate is more in human babies born with more pounds or less than 8 pounds. So that's why nature eliminate the human babies born with less than 8 pounds or more than 8 pounds. So finally what happened? Nature select the human babies born with 8 pounds. So only one phenotype, average phenotype. Due to operation of stabilizing selection, no new species is evolved. The existing genotypes stabilized due to operation of or due to function of stabilizing selection. Stabilizing selection, also known as normalizing selection, it select the average phenotypes. That's why stabilizing selection, also known as centripetal selection. Centripetal selection. Centripetal selection. Stabilizing selection, also known as normalizing selection. Stabilizing selection is operating in stable environmental condition. Stabilizing selection select the average phenotype, eliminate the extreme phenotype from both the ends. As a result, peak become narrow and higher. So eliminate the extreme phenotypes. As a result, what happen? Average phenotype number increases. For example, average phenotype 80, one extreme phenotype 10%, another extreme phenotype 10%. So it eliminates 20%. So now the average phenotype when compared to other extreme phenotypes increases. So peak become narrow and higher. Means stabilizing selection select the average phenotype. That's why stabilizing selection, also known as centripetal selection. One example is in England, birth weights of human babies were estimated in large scale. Human babies born with 8 pounds is average phenotypes. Human babies born with more than 8 pounds, one extreme phenotype. Human babies born with less than 8 pounds, another extreme phenotype. Mortality rate is more in human babies born with more than 8 pounds or less than 8 pounds. So mortality rate is negligible in human babies born with 8 pounds. So that means nature or stabilizing selection select the human babies born with 8 pounds. So stabilizing selection. Now we discuss directional selection. Directional selection. Direction selection. Direction selection is operating when gradual changes occur in the environment. At the time, it selects one extreme phenotype. As a result, mean shifted to other end. Means peak shifted to other end. Peak shifted to other end. So before operation of directional selection, peak is normal phenotype, means average phenotype. Most of the individuals, that is the average phenotype. For example, 80 normal neck giraffe, 10 short neck giraffe, 10 long neck giraffe. So directional selection is operating when gradual changes occur in the environment. These changes favor the long neck. So directional selection select one extreme phenotype. So average is shifted to that end. Previously average is a normal neck giraffe. So due to operation of direction selection, peak shifted to another end in most of the individuals possess long neck. So long neck become average phenotype. So now two extreme phenotypes are, one extreme phenotype short neck giraffe, another extreme phenotype normal neck giraffe. So once the neck size is sufficient to reach the greater foliage, instead direction selection, stabilizing selection is operating. You already know, due to operation of stabilizing selection, extreme phenotypes are eliminated. In this population, extreme phenotype is short neck and normal neck giraffe. So short neck and normal neck giraffe completely eliminated. Now in that population, only long neck giraffe. So due to operation of direction selection followed by stabilizing selection, one new species is evolved. Due to functioning of stabilizing selection alone, no new species is evolved. So due to functioning of direction selection followed by stabilizing selection, one new species is evolved. Once again, direction selection. When gradual changes occur in the environment, at the time direction selection is operating. Direction selection select one extreme phenotype. As a result, peak shifted to other end. Previously, peak is average phenotype. Due to operation of direction selection, one extreme phenotype become average phenotype. Once the average phenotype perform a particular function effectively in that population, instead direction selection, stabilizing selection is operating. As a result, new species is evolved. Now we discuss the example. So 100 giraffe, for example, 100 giraffe are living, 80 giraffe had normal neck, 10 giraffe had long neck, 10 giraffe had short neck. Short neck and long neck extreme phenotypes. Gradual changes in the environment. So grasses are, due to drought condition, grasses not available. They forcefully depends upon the foliage. Due to presence of long neck, so they reach the lower branches of the trees. In this manner, they obtain the food. So direction selection select the extreme phenotype, that is long neck giraffe. Previously, average phenotype is normal neck. As a result, peak shifted, mean shifted to that end. Previously, normal neck giraffe, average phenotype. Now, long neck giraffe become average phenotype. Normal neck and short neck giraffe become extreme phenotypes. Once the neck size is sufficient to reach the greater foliage, now instead directional selection, stabilizing selection is operating. The stabilizing selection eliminate the normal neck and short neck giraffe. In the population, only long neck giraffe present. In this manner, due to functioning of direction selection followed by stabilizing selection, one new species is evolved. Now we discuss disruptive selection. disruptive selection, also known as centrifugal selection. Disruptive selection or centrifugal selection. It is operating in heterogeneous environment, means at present homogeneous environment. The existing environment is suitable for average phenotype. Now the homogeneous environment become heterogeneous. So the heterogeneous environment is suitable to extreme phenotypes and not suitable to average phenotype. That means disruptive selection select all extreme phenotypes, eliminate the average phenotype. And each extreme phenotype is known as subpopulation or species population. Still, members of one subpopulation reproduce with members of another subpopulation in course of time. So each subpopulation reproductively isolated from another subpopulation. At the time we can say each subpopulation as a new species. In this manner, due to operation of disruptive selection, more than one new species is evolved. The rare type of selection, disruptive selection, select the extreme phenotypes. That's why it is known as centrifugal selection. That's why in your textbook author mentioned due to operation of disruptive selection, two peaks are formed. Two peaks are formed. So peak means here extreme phenotype. So number of peaks depends upon number of extreme phenotype. Two peaks are formed and each one is known as subpopulation. In course of time, members of one subpopulation reproductively isolated from members of other subpopulation. At the time, each subpopulation considered as a new species. So disruptive selection, also known as centrifugal selection, it is a rare type of selection. A rare type of selection. Disruptive selection, select the all extreme phenotypes, eliminate the average phenotype. So each extreme phenotype is known as subpopulation or species population. Once the members of one subpopulation reproductively separated from other subpopulation, each subpopulation considered as a new species. From one sunflower population in California, two subpopulations are evolved. One subpopulation live in dry area, another subpopulation live in wet area. Similarly, evolution of finch birds, example for disruptive selection. From seed-eating finch bird, seed-eating finch bird is the average phenotype and 20 extreme phenotypes. So the disruptive selection eliminated the seed-eating finch birds. So as a result, 20 finch bird species evolved. Example for disruptive selection. Similarly, in peppered moth, one subpopulation is white color, another subpopulation is black color. Now they are not reproductively isolated. In future they are reproductively isolated. So in future each color means white color and black color evolved into two different species. Example for disruptive selection. So due to operation of disruptive selection, two peaks are formed, means two extreme phenotype, two species minimum. Due to operation of directional selection, peak shifted to another end. So due to operation of stabilizing selection, peak become narrow and higher. Next one is mutation. Due to mutation also new alleles are formed. Once new alleles are formed, change occur in the allelic frequencies, change occur in the genotypic frequency. Before 1900, in Drosophila only red color, eye color is red. After 1900, due to mutation, white color. Before 1900, all Drosophila had red eyes, maybe male and female red eyes. After, due to mutation, new allele is formed. Now, capital smaller, smaller, capital smaller. So before industrialization, before 1900, in Drosophila, all Drosophila had red eyes. In female, only one genotype. The allele for eye color present on the X chromosome. All females red eye, all males red eye. After mutation, new allele, smaller allele is formed. That's why females 3 genotypes, males 2 genotypes. So before mutation only 1 genotype. So in this manner, due to mutation also allelic frequency and genotypic frequency changes. Due to change in the allelic frequency and genotypic frequency, a new species is evolved. So next class we discuss brief account of evolution of human being and PU questions. So in the next class I complete this chapter, evolution.